All right. Hi, everyone. Yeah, as I said, my name is Aaron Purdy. I'm another one of the Marine Mammal Research Unit's master's students. And for this past year, I've been studying diving physiology in Steller sea lions. This is one of those Steller sea lions right there by the name of Sitka. So for this presentation, I hope to give you just a brief overview of what I've been doing over the past summer and some of the goals that I hope to accomplish by the end of my master's. I also hope to convince my supervisor that I haven't been completely wasting his and everybody else's time. We'll see if I can do that. So first off, a little overview of why studying Steller sea lions is important. So as I'm sure many of you know, from the late 1970s to the late 1990s, there's been a huge population decline in particularly the eastern population of Steller sea lions along the Gulf of Alaska in the Aleutian Islands, kind of where at the bottom of those red dots right there, we saw declines of more than 80% in some areas. So trying to figure out why some of these declines are happening. One possibility that might be causing this is that there's been a shift in prey. So it might be that these animals are no longer able to meet their daily energetic requirements. And because of that, it's really important that we understand their foraging capabilities. So understanding how well these animals are able to dive, so we know what happens when they're pushed to their limits, which might be happening in the wild. So one of these limits is called the aerobic dive limit. In the diving physiologist world, this is, people are kind of obsessed with it, but it is for good reason. The aerobic dive limit is defined as the point in a dive in which you begin to accumulate lactic acid in your system. And the reason you do this is you're switching from aerobic metabolism to anaerobic metabolism. It means you're using up your oxygen stores, you're reaching the ends of your limits right there. Diving past this point is associated with different costs. And you can kind of break these costs into two different groups. First is an energetic cost. So it takes energy to rid the body of these metabolites that are building up in your system. And the second is a time cost. So the only time in which you're able to actually rid your body of these metabolites is while you're at the surface breathing oxygen. If you're at the surface, that means you are not foraging. So if with an increased recovery time, you're going to have less time to eat, essentially. So what I'm interested in is looking at how recovery time is related to this aerobic dive limit. So this is something that I kind of expected, one of my predictions when I was going out with beginning my research, that with an increase in dive time, you're going to have an increase in recovery time. That just makes sense. The harder you work, the more time you have to take to recover. But I was expecting once you surpass that aerobic dive limit, you're then going to have a disproportionate increase in recovery time because you have to spend that much more time recovering from all of these metabolites accumulating in your system. So my research goals, I had two main things I wanted to set out to do. Was one, determine the aerobic dive limit in Steller sea lions. This hasn't been done in very many pinnipeds at all, and so this is something that'd be really interesting to see. And then also looking at how recovery time changes with increased dive effort. Another way of putting that is seeing how recovery time changes after they exceed that aerobic dive limit. So how do you do that in a bunch of sea lions? I've been lucky enough to work with, in partnership with the Vancouver Aquarium at the Open Water Research Station. Out there they have 4 trained Steller sea lions. You saw one at the beginning, that was Sitka. This one here is Bonnie. You can tell by those beautiful long whiskers. She's adorable. These animals are incredibly well trained. They're actually able of swimming, they're trained to swim out in the open ocean and perform dives out there. And so we're actually able to take these animals into a more natural environment to see how these animals are going to be functioning in the wild. On the right, you can see a picture of the setup that we have out there. We have a floating barge where we do most of our data collection, and I'll give you an overview of how that works. So first off, I'll show you kind of the setup that we had for measuring lactic acid. We have this floating barge, you can see in those gray boxes there, with fish pumps these tubes that extend down up to 40 meters into the water, so down to relevant foraging depths that you'd see sea lions using in the wild. And like the name implies, fish pumps, we can pump fish down through them. So this encourages the sea lions to dive and then stay in that location. So sea lions dive, they're swimming back and forth between these two tubes, and they're feeding on the fish. It mimics a foraging bout. When the fish stops, they don't see no point in staying there anymore, so they surface, and the trainers then have them come up onto the barge, where we take a voluntary blood sample, and then we use just a small handheld lactate meter, similar to what some people, if you've ever had someone who's training for running, they use something similar to this. They get a small blood sample, and we can measure the lactic acid in their blood. So this is what it actually looks like when you're out on the water doing this. And right now, this looks kind of like a calm day, but if you can imagine, you're out on a floating dock, it's rocking back and forth, there's wind, there's rain, there's boats driving past. It's incredible that these trainers are able to get this blood from these animals, particularly because they're completely wide awake the entire time. They are voluntarily giving that blood sample. It just shows how great this connection these trainers have with these animals, and just how much work they've been able to put into it. It's really fantastic. So then that is how we measured lactic acid. Another part of the study was measuring that recovery time. So a similar setup where the sea lions dive down, they're feeding on fish, but when the fish stops, they then, instead of surfacing and coming up onto the barge, they instead surfaced underneath this metabolic chamber. It's a sealed chamber where we have air flowing over them. They're breathing in oxygen, they're breathing out CO2, and we can get measurements of both of those gases and use that to measure their metabolic rate. So after a dive, their metabolic rate is going to be increased. We're measuring how long it takes for them to get back to that normal level, determine how long it takes for them to recover from that dive. So things that I still hope to do, I'm going to analyze that recovery time for oxygen and CO2 and see how they interact with that aerobic dive limit. Then hopefully I'm going to be able to use all of these together to model different foraging strategies that these animals might be using in the wild. So hopefully I've been able to convince you that it hasn't all been fun and games out there. I have been doing some work despite what looks there might be. A big thank you to all of the staff at Open Water and to all my other support. If you have any questions, please ask. Thank you.
Do you have any questions?
The main question, I assume, is working with sea lions that cool? And yeah, it is.
Yeah, we'll let you off the hook with that. Thanks, Aaron.